Welcome to the first video of Biochemistry 1. Here in Chapter 1, we're just going to do some review. We're going to do some review of basic chemistry, some basic biology, and basic physics. Hopefully things you've covered before in your previous classes. If you're not sure of anything we cover in these introduction videos, please come and see me at any point, or please look back at your previous courses that you've done just to refresh yourself. But we're just going to look at a very basic overview of chemistry to begin with in section 1.1. And the key concepts for us to learn are that biological molecules are only made out of a certain number of building blocks. And biochemistry is really dictated between what groups are on a biomolecule and what types of linkages these groups form. We're also going to look at the idea of evolution, how we got from small molecules towards life, very, very briefly. And we're also going to introduce the concept that molecules can self-replicate, and that's really how we have life and how that relates to evolution. All right, so let's just look at the elements that make up living systems. Now, unlike general chemistry or organic chemistry, in biochemistry, we really limit the periodic table. If you take a look, you can see that in dry weight, that carbon is the most abundant. 61% of the weight of organisms is due to carbon. So in biochemistry, we really focus on carbon chemistry. The next two abundant molecules are nitrogen and oxygen, which again are very important when it comes to chemistry. And then we have a lot of ions, a lot of positive ions, cations. And these will be important when it comes to catalysts and other types of reactions that happen in the body. Note that this is a dry weight. That is, we don't consider the water in living organisms because if we considered water, then oxygen would most likely be the heaviest element since we're mostly water, but we're only dry weight. And talking about life, life is incredibly old. The oldest known fossil, if you will, of life is shown here, and it's 3.5 billion years old. Billion with a B. So life has been around for longer than really any human can contemplate, where we think 10,000 years is a extremely long time, while 10,000 years is nothing in terms of the Earth, not even a second. Right? So where did life come from? Where did chemicals come from? And there's a lot of theories out there. One of the popular theories in the earliest 20th century was that either UV light or lightning created the first organic molecules. This was pushed by Oberon and Haldane in the 20s. And in 1953, Miller and Urey actually did an experiment where they tried to replicate early Earth's atmosphere, gave it an electric shock, and they actually got some amino acids out of it, showing that it is plausible. Other scientists believe that thermal vents are where our first organic molecules came from. And the reason is that they're 400 C. This is supposed to be a degree marker. So thermal vents extremely hot, 400 C, and from these thermal vents you get these metal sulfur complexes. And you can think that it's hot enough, and if there's enough material around, that you could create the early amino acids from these vents. And if we look at vents now, we see that there is life around these vents, so it is not implausible that the early molecules of life came from these vents. Now, having just organic molecules is great, but you can't have life unless they can come together. And so the key reaction we think about when it comes to life or biomolecules is condensation and hydrolysis. So if you combine two things together, and you lose water, that is called a condensation reaction. You have created one thing, and it's condensation because you lose water. While if you take one thing and you cut it with water, that's called hydrolysis. So condensation, water is a product. Hydrolysis, hydro, water, lysis, cutting. You are cutting something apart using water. I do want you to remember lysis means cut, because in biochemistry we see the word lysis a lot. It's going to appear a lot, and it always means the same thing. It always means cutting. So before there was life in the prebiotic era, we had these molecules, these building blocks, and they had to come together somehow, and that was usually through condensation. Now, like I said, in biochemistry, we really care about what groups are on our biomolecules. And since we only use a certain number of compounds, elements, carbon, nitrogen, oxygen, and hydrogen, we only get a certain amount of functional groups. And here's a table of the functional groups. Now, I would like you to know these functional groups. And by know, I mean you should be able to look at these and name them. Or if I give you a name, you should draw them. And like I said, if you know how a functional group interacts chemically, you can begin to understand how a biomolecule, which is made out of functional groups, can interact. And a lot of these compounds are actually complementary to each other. For example, an amino group is charged, NH3, while a carboxylic group is negatively charged, C-double-O. And this complementary nature can lead to replication, because let's say you have two strands. If these two strands are complementary to each other, well, you only need one strand to determine what the other strand looks like. And we'll get into this topic more in depth in the later chapters. Here are the type of bonds you can really have in biochemistry, where amide, disulfide, phosphoester, so on and so forth. Not much to say about this table. Again, something I'd like you to know, though, be able to recognize and name these compounds. Now, in biochemistry, we aren't necessarily interested in the functional groups. We should know them, and we should know how they work, but we really care about what they create. And when these functional groups come together, they create what are called polymers. Polymers are made out of monomers, so one segment of a polymer is called a monomer. Many monomers together are a polymer. And the polymers that we really care about are amino acids, protein, nucleotides, nucleic acids, monosaccharides, which are polysaccharides, and one more that's not listed, which are lipids, which are made out of fatty acids. And this idea complementary is shown here in this image. So DNA is the best example. DNA has 4 bases, A, C, G, and T. These pair with T, G, C, and A. So when you have one polymer that breaks apart or unfolds, and this is as shown here, you can know what the other side should be just by matching. So for here, cream goes with purple. So if one strand is a cream, 3 creams and 3 purples, the other strand must be 3 purples and 3 creams. You have just replicated yourself. And that's it for our first video. I will see you in the next video.